Alright, so the purpose of this video is to show you how lysosomes are formed and to show you how two different types of proteins are formed. The hydrolytic enzymes that are formed inside the lysosome and the ATPase proton pump that is part of the membrane of the lysosome. Okay, so I have two ribosomes here and they're located on the ER. This ribosome is going to make the ATPase, and this ribosome is going to make the hydrolytic enzyme. So the hydrolytic enzyme, so if the mRNA is here for the hydrolytic enzyme, the enzyme is going to be produced inside the space of the ER, which is called the lumen. But for ATPase, it's going to actually produce it in the membrane. There. Okay, so this protein here, this is a hydrolytic enzyme. The membrane will then bulge out and the enzyme will go inside of a vesicle. And that'll be transported via the motor proteins on the cytoskeleton to the Golgi. So when the Golgi body and this membrane fuse, you'll end up with. So the reason why this is called the endomembrane system is because the membranes can be interchanged. So this membrane here became part of that Golgi membrane, and then here's that hydrolytic enzyme now inside the Golgi body. While it was in the ER, there was a sugar added to the outside called mannose. And mannose is a type of sugar. And so the mannose would be here as well, already on the protein, on the enzyme that's in here. But inside the Golgi body, a phosphate group is attached. So now this is going to be mannose 6-phosphate. So all of the hydrolytic enzymes that are found in the Golgi body are tagged with the mannose 6-phosphate tag. And that is going to be really important as far as how it ends up in the Golgi and not somewhere else. Okay, so then this protein can then end up budding out of the Golgi. So in order for it to bud out, there's a little receptor on the inside of the membrane that binds to mannose 6-phosphate. So since this enzyme has mannose 6-phosphate on it, it'll stick there. There's also another protein inside this membrane, and that was here, our ATPase. So when this membrane protein formed a bubble, let's say it formed it here, the ATPase would be part of that membrane, and when that vesicle fused here, it would be part of the membrane of the Golgi body, and that's how it could end up as the ATPase here, because all these membranes can be they're all fluid and they can kind of move around. So now we have a vesicle that's forming here that has two proteins in it. Of course, lysosomes have lots of enzymes in them, and I'm just showing you how one of them is in there, but there would be more than one enzyme coming in here. So now this is a bubble, right? This is what's called an early lysosome. And it has already an ATPase, and it has an enzyme with a mannose 6-phosphate tag, so that would be one of the hydrolytic enzymes. Okay, of course there'd be a lot of hydrolytic enzymes. So an early lysosome is not quite mature. So what has to happen is early lysosome has to fuse with a phagosome. So a phagosome happens when the cell membrane buds inward. And usually that happens because it's doing endocytosis. But on the membrane of the cell membrane are already ATPases. Okay, so, and notice that this is a little light bulb facing out, and this is a little light bulb facing in, because when this forms when this buds inward, what will happen is right? You'll get that ATPase with the little bulb facing in. So when this makes a bubble, the ATPase is facing inward. And of course, there could be many of those. And there's other membrane proteins too. Now, there could be something inside here that the lysosome is going to break down, but that's not really important for what I'm going to show you here. And that is that these two bubbles fuse to make a mature lysosome. So this is called an endosome, or a phagosome. And together, when they fuse together, you're going to get the mature lysosome, which will have half of the membrane from here and half of the membrane from here. But when they fuse together to make one big bubble, I mean, it doesn't really matter that there's the origins of the two membranes are different. But you get the mature lysosome. I'm off the page there a little bit. Okay. And so you have your hydrolytic enzymes with the mannose 6-phosphate tags, and then you have all these ATPases facing inward. And what they're going to do is they're going to use ATP, and that's going to phosphorylate this enzyme. And it's actually a little motor which pumps protons inside the lysosome, and that's going to reduce the pH in here and activate the enzymes that are found in the lysosome. So these ATPase proton pumps are really important to getting the lysosome to function.